Hello everyone, welcome back to our channel. In today's lecture, we will discuss biosimilar substitution and regulatory pharmacology, an important topic in modern clinical practice. Biologic medicines have transformed the treatment of many chronic and life threatening diseases. As these medicines become more widely used, biosimilars provide a more affordable treatment option while maintaining comparable quality, safety, and effectiveness. However, unlike generic medicines, biosimilars are not exact copies of the original biologic because they are produced using the living cells. Therefore, their approval, substitution, and interchangeability require strict regulatory evaluation. In this lecture, we will understand what biosimilars are, how they differ from the generics, the regulatory approval process, substitution policies, and their importance in the clinical practice. So, let's begin by understanding the basics of biosimilars. Let's understand this concept with a clinical case. Meet Mrs. Priya Nair, a 42-year-old woman diagnosed with rheumatoid arthritis for past 5 years. She has been receiving adalimumab, also known as Humira, 40 mg subcutaneously every 2 weeks for the last 3 years. She uses a self-injection pen and is currently in stable clinical remission. Now, due to an insurance policy, her medication was switched to a biosimilar without consulting the prescribing doctor or confirming whether the product was interchangeable. Naturally, she became concerned and questioned whether biosimilars are as effective as the original biologic. However, her laboratory findings are reassuring. Her DAS28 score is 1.8, indicating remission. Therapeutic drug levels are maintained. Anti drug antibodies are negative, and inflammatory markers CRP and ESR remain within the normal limits. This case highlights the importance of understanding biosimilar substitution, regulatory approval, and appropriate clinical decision making. Now, let's discuss the most important clinical decision in this case. After switching to the biosimilar, Priya reports that the new medicine feels different. As a healthcare professional, what should be the first step? Option A is not appropriate because simply reassuring the patient and assuming it's a nocebo effect may overlook the genuine clinical concerns. Option B is also not the best choice because switching back to the original biologic immediately without proper evaluation is a premature decision. So the correct option is C. Verify, assess, and then decide. First, confirm which biosimilar was dispensed and whether it has regulatory approval for interchangeability. Assess the patient's clinical condition and the laboratory findings, and ensure the substitution complies with local regulations. Only after a complete evaluation should any treatment decision be made. The patient centered and evidence based approach ensures both treatment effectiveness and patient safety. Now, let's understand a common misconception known as the generics fallacy. Many people think that biosimilars are same as the generic medicines, but this is incorrect. Generic drugs are chemically identical to the original small molecule drug. So, in many cases, they can be substituted automatically. However, biologic medicines are produced from the living cells and are highly complex molecules. Therefore, biosimilars are highly similar but not identical to the reference biologic. Because of this complexity, automatic substitution is not allowed unless specific regulatory requirements such as interchangeability are met. Substitution can fail when stable patients are switched off for non medical reasons, when the product traceability is not maintained, or when the healthcare professionals assume that the substitution rules are same worldwide. In reality, regulatory agencies like the FDA, EMA, and CDSCO have different guidelines for biosimilar substitution. So the key message is simple. Generics can often be substituted automatically, but biosimilars require careful clinical and regulatory evaluation before substitution. Let's understand how a biosimilar is approved by the regulatory authorities like the US FDA through the 351K Biologics License Application or BLA pathway. Unlike generic drugs, biosimilars are approved using the totality of the evidence approach. This means that no single study is enough. Instead, the scientific evidence is evaluated together. The first pillar is structural and analytical similarity, where the scientists compare the amino acid sequence, three dimensional structure, glycosylation pattern, and other physiochemical properties with a reference biologic. The second pillar is functional and biological similarity, which evaluates receptor binding, biological activity, Fc mediated functions, and pharmacokinetic and pharmacodynamic profiles. Finally, clinical similarity is assessed through comparative clinical studies, including the pharmacokinetics, immunogenicity, and safety, to confirm that there are no clinically meaningful differences between the biosimilar and the reference drug. This rigorous stepwise approach ensures that approved biosimilars are safe, effective, and of high quality for the patient use. This comparison matrix clearly shows that the differences between the generic drugs, biosimilars, and interchangeable biosimilars. First, generic medicines are chemically identical to the reference drug and are approved through the ANDA 505J pathway. They can usually be substituted automatically at the pharmacy. In contrast, biosimilars are highly similar but not identical to the original biologic. They are approved through the 351K Biologics License Application pathway and generally cannot be automatically substituted. Clinical evidence is required to demonstrate the comparable safety, efficacy, and the quality. An interchangeable biosimilar is a special category. In addition to meeting biosimilar requirements, it must provide switching study data showing that the patients can safely alternate between the reference product and the biosimilar without additional risk. Depending on the local regulations, these products may be substituted at the pharmacy. So, the key takeaway is that all the interchangeable biosimilars are biosimilars, but not all biosimilars are interchangeable. Understanding this distinction is essential for safe prescribing and dispensing. Let's understand what makes a biosimilar eligible for the interchangeability under the FDA standard. To receive this designation, manufacturers must conduct a switching study where the patients alternate multiple times between the reference biologic and the biosimilar. The goal is to demonstrate that switching does not affect the treatment outcomes. The study also evaluates pharmacokinetic equivalence, ensuring that the drug exposure measured by the AUC and Cmax remains within the acceptable range. In addition, researchers assess immunogenicity to confirm that switching does not increase the formation of the anti-drug antibodies. In these studies show efficacy, safety, and immunogenicity, they remain comparable. The biosimilar may qualify as interchangeable. However, it is important to remember that even an interchangeable biosimilar must still follow local pharmacy and regulatory laws before it can be substituted without the prescriber's approval. Now, let's compare how different regulatory agencies across the world approach biosimilar approval and substitution. In the US, the FDA approves biosimilars through the 351K pathway. Products are classified as either biosimilars or interchangeable biosimilars, and only interchangeable products may be eligible for the pharmacy level substitution according to the state laws. In the European Union, the EMA European Medicines Agency evaluates biosimilars, but the decision regarding the automatic substitution are made by the individual member countries. There is no centralized EU designation for interchangeability. In India, the CDSCO, Central Drugs Standard Control Organization, regulates biosimilars under the Similar Biologics Guideline. Automatic substitution is generally not recommended, and switching should occur under the supervision of the prescribing physician. India also requires robust clinical evidence before the approval. So, Biosimilars are evaluated globally using high scientific standards, but the substitution policies differ from one country to another. Therefore, healthcare professionals should always follow the regulations applicable in their own country. This slide summarizes the golden rule of the biosimilar substitution. A biosimilar should not be substituted without the prescriber's knowledge unless it has an interchangeability designation, and such substitution is permitted under local regulations. The four important steps to follow are: first, verify the product's interchangeability status using the official regulatory databases such as FDA Purple Book or your national regulatory authority. Second, confirm the legal requirements in your own country or region regarding the pharmacy level substitution and prescriber notification. Third, document the exact product name, manufacturer, and batch or lot number and communicate any substitution to the prescribing physician. This is essential for proper pharmacovigilance. Finally, monitor the patient's clinical response and report any adverse events or unexpected changes in efficacy or safety. So, the ultimate goal is safe, evidence based substitution while ensuring the continuity of the patient care and regulatory compliance. Now, let's discuss two important reasons why patients may experience problems after switching to a biosimilar: immunogenicity and nocebo effect. On the left is the immunogenicity, which is a biological risk. Sometimes the immune system recognizes the biologic drug as foreign and produces anti drug antibodies or ADAs. These antibodies may bind to the drug and reduce its concentration, or they may neutralize the drug, decreasing its therapeutic effect. Laboratory tests such as ELISA are commonly used to detect these antibodies. On the right side is the nocebo effect, which is a physiological phenomenon. Here, patients develop negative expectation after switching to a biosimilar and may report worsening symptoms even when the objective clinical findings remain unchanged. Therefore, when the patient reports new symptoms after switching healthcare, professionals should evaluate both biological causes such as immunogenicity and physiological factors such as nocebo effect before making any treatment changes. This balanced approach helps ensure appropriate patient care and avoids unnecessary discontinuation of the effective therapy. Let's understand another important concept in biosimilar regulation: extrapolation of the indications. Consider this example. A biosimilar anti TNF monoclonal antibody has been clinically tested and approved for rheumatoid arthritis. The original difference and the reference biologic is also approved for Crohn's disease. The question is, can the biosimilar also be approved for the Crohn's disease without conducting a separate clinical trial? The answer is yes, but only if there is a strong scientific justification through extrapolation. Regulatory agencies evaluate three factors. First, the mechanism of action must be same in both the diseases. Second, the safety and the immunogenicity profile should remain consistent across the indications. Third, all supporting scientific evidence must be carefully reviewed and accepted by the regulatory authority. So, remember that the extrapolation is a scientific and regulatory decision, not an automatic approval. It allows biosimilars to be approved for multiple indications while avoiding unnecessary duplication of clinical trials, provided the evidence is robust. After a biosimilar is approved, continuous safety monitoring becomes extremely important. This process is known as the pharmacovigilance, and one of its key principles is traceability. Traceability means that the every adverse event can be linked to the exact biological product, including its brand name and the batch or the lot number. This helps to identify the specific medicine responsible for any safety concern. Different regulatory agencies use different monitoring systems. The FDA uses MedWatch and FAERS FAIRS, while the European Medicine Agency monitors safety through the UDRA vigilance. For biologics with important safety concerns, additional risk management programs such as REMS may also be required. Another valuable source of information is real world evidence, which collects safety and effectiveness data from post marketing registers and electronic health records. The key message is that approval is not the end of the process. Continuous pharmacovigilance and accurate traceability are essential to ensure the long term safety and effectiveness of biosimilars in the clinical practice. Finally, let's discuss situations where a biosimilar should not be substituted. These are important red flags that every healthcare professional should recognize. First, if the biosimilar does not have an interchangeability designation in that particular country or region, it should not be substituted automatically. Second, avoid switching if the parent is clinically unstable with the patient or experiencing an active disease flare. In such cases, it becomes difficult to determine whether the worsening symptoms are due to the disease itself or the medication switch. Third, if the patient has a previous adverse reaction such as anti-drug antibody formation, hypersensitivity, or an infusion reaction to that specific biosimilar, substitution should be avoided unless carefully evaluated by the treating physician. Finally, substitution should not occur if proper traceability cannot be maintained. The product name, manufacturer, and batch or lot number must always be documented for effective pharmacovigilance. So, the key takeaway is that patient safety always comes first. When these red flags are present, avoid automatic substitution and make decisions based on the clinical judgment, regulatory guidance, and individual patient needs. Now, let's conclude by the Indian perspective on biosimilars and their use in the clinical practice. In India, biosimilars are regulated by the SADSCO under the Similar Biologics Guideline. The approval process requires comprehensive quality, non-clinical and clinical data, including phase 3 clinical trials for many products to ensure the safety and the efficacy. An important point to remember is that automatic substitution of biosimilars is not recommended in India based on the decision to watch from the reference biologic to a biosimilar should be made by the treating physician based on the patient's clinical condition. For better traceability, biologics should ideally be prescribed by their brand name, allowing accurate identification of the product and batch number if any adverse event occurs. Healthcare professionals should also report suspected adverse drug reactions through the Pharmacovigilance Program of India or PVPI to support continuous safety monitoring. Finally, biosimilars have significantly improved access to advanced therapies by reducing the treatment cost, making biologic medicines more affordable for many patients across India. So the key takeaway that biosimilars offer a safe and cost-effective treatment option when used according to SADSCO guidelines with appropriate physician oversight and ongoing pharmacovigilance. Let's conclude today's lecture with the key messages. Remember, a biosimilar is not same as the generic drug and not every biosimilar is interchangeable. Before any substitution, healthcare professionals must consider the regulatory designation, clinical evidence and the patient's condition. Safe use of the biosimilars depends on the scientific evidence, proper traceability, pharmacovigilance and informed clinical decision making. These principles help ensure both treatment effectiveness and patient safety. I hope this lecture has helped you to understand the concepts of biosimilar substitution and the regulatory pharmacology in simple and practical way. Thank you so much for watching this lecture. I hope this session has given you a clear understanding of biosimilar substitution, interchangeability, regulatory pharmacology, and their importance in clinical practice. If you found this lecture helpful, please like the video, share it with your friends and colleagues, and subscribe our channel for more high quality lectures on MBBS, Pharm.D, pharmacology, clinical pharmacy, and medicine. If you have any questions or topic suggestions, feel free to leave them in the comments below. Your feedback helps us create better educational content. Thank you once again for your support. Keep learning, keep growing, and I'll see you in the next lecture. Take care.